Hello students, welcome to LEELA'S tutorial. So today we are going to continue the last class, that is conservation of germplasm. In that you are going to conserve the germplasm by different ways. In that one of the type was cryopreservation. Okay, so cryopreservation, the name itself implies that something is preserved at very low temperatures. Cryo means low, right? So cryo, or you can say the freezing temperature. Okay, so how you define it? It is a process of preserving the cells or tissues at very low temperature, right? That is under freezing condition. So here the cells are maintained as the frozen cells or tissues. So let us see in detail what are the steps involved for this complete process. So first step is selection of cells or tissue. If you want to conserve a germplasm, you will be selecting the tissue, the germplasm or the endosperm, right? So here first you are going to select whatever the cells or the tissues you are going to store it. Okay, so then the second step is pre-treatment. So here before entering into the storage or the preservation process, you are going to make the cells tolerant to the new environment. So here what will be the new environment? As it is a long-term storage, you are going to add the preservatives. So the cells must be tolerant to that preservative, and also you are maintaining them at very low temperatures, that is in frozen condition. So here again you have to make them cryo-tolerant, right? So here first it is a pre-treatment where you are making them tolerant to the toxic preservatives, or you are making tolerant to the frozen conditions. Okay, so once the cells are stable, you are going to add your cryopreservant, right? The chemical substance which is used for cryopreservation process. So now the cryopreservant, it can be your sucrose, right? Alcohol, glycols or DMSO, right? Dimethyl sulfoxide. Any two can be used as cryopreservant. Okay, so when you use any two, here then next step will be the freezing. So after using or after adding the cryopreservant, any two from these, when you add it to the pre-treated tissue, what happens? You have to maintain the tissue in cold conditions, right? You are not going to freeze them directly. You are going to place them in low conditions. Gradually you are going to decrease the temperature, right? So then till it reaches to the freezing. So once you get the freezing or frozen cells, you are going to store them in liquid nitrogen where it is maintained at minus 196 degrees centigrade. Okay, the liquid nitrogen tanks, you are going to place this container, or you can say place the tissue along with the cryopreservant in a vials and allow them to undergo freezing, and then you are going to store them in liquid nitrogen tanks. Okay, so after that, now this storage can be for more than 10 or 20 years. Okay, so it depends on the tissue and the conditions. Once you store them, now after certain time period, you require that tissue. That means you have to use that tissue again. You want that plant to be produced again. So what you do? You have to revive those tissue. So here, up to here you have stored it for few years. Now after certain time period, you want the tissue to again produce a plant. In that case, you have to go for the reviving process. So here when you are freezing, what is happening? All the liquid content of the cell is converting into ice crystals, and also the enzymes which are present in the cells are losing their activity. That means the cells have lost their metabolic activity, but they are in live condition. Is that clear? So now you want these cells to become again active, or you can say metabolically active. In that case, what you have to do? The frozen content of the cell, the liquid content and the enzyme activity has to be revived. Again, they has to be revived. So how you can revive them? Simply either by placing them in a normal room temperature for longer time. Otherwise you can use warm water baths. Right? The warm water you are going to place those vials. Okay? Directly you are not going to place them in hot water. Again, because of sudden shock, right? The temperature change, the cells may lose their viability. Okay? So here, then next step is thawing process. Otherwise, you can either maintain them in warm water baths or you can thaw them. Thaw means it is simply a swallowing process. So you are going to place a vial and simply thaw them, so that the heat generated will be making them revived. Okay, the crystals will be melted and you get the cells with their enzymes in active condition. So now once you have got the cells, the revived cells, now you have to go for the washing process. So the washing process, simply by introducing the buffers or the water, you are going to wash the preservatives whatever you have added. Right? Simply rinse them with the micropipette, right, within the vial, and then you are going for the reculturing process. So when you wash them, you are going to add new media. Right? New tissue culture media or the growth media will be added to those vials. So after that washing, you are going to add the new media and allow them to undergo subculturing process, or you can say it as reculturing. That means the frozen cells are revived and those cells are allowed to grow in new media. Okay, so now in this media in this culture, there may be cells which might have died because of either the toxic activity or due to the sudden change in the temperature. So what you do? You go for the measurement of viability, or you can say the viability measurement or viability testing must be done. So after the reculturing, you are going to use this formula, that is the number of cells growing, that means after subculturing or reculturing, how many cells have been increased, divided by number of cells present after thawing. Right? After thawing, what is the number of cells, and after reculturing, what is the number of cells, into 100. Then you get the actual viable cells. Only viable cells or the live cells can divide. Right? If the cells are dead, they can't die. So based on this, you can get what is the number of cells which are live and which are dividing. Is that clear? So once you get the viable cells and subculturing process, then you can go for your regeneration of the plants by using your plant tissue culture media. Is that clear? So now after this complete process, okay, when you regenerate the plant after so many years, again you are going to check for the any variations. Okay, maximum there will not be any variation as it is preserved under minus 196 degrees centigrade, very low temperatures. Okay, there is no metabolic activity of the cell. Okay, so here the metabolic activity is lost, and here again it is gained, but the cell is in live condition only. Okay, so let us see now what are the different types of cryopreservation. Here I told they are stored in minus 196 degrees centigrade. So what are the different conditions in order to store them? So here it is classified into 4 types. First one is slow freezing method. So why slow freezing method? Because the cells may die of freezage, right, or freeze shock or cold shock. In order to avoid that, what you do? You gradually cool the cells or you gradually decrease the temperature so that they enter into the frozen state. Okay, once they are frozen, it is a partially dehydrated cell. It is not completely dried or a powder like lyophilization. Lyophilization is different from cryopreservation. So here you can say that it is partially dehydrated tissue. And this tissue, if you go for the slow freezing method, the rate of survival is very high. That means the viable number of cells will be high in this case. Okay, the damage to the tissue or the cells caused is very low. Then coming to the second method, it is rapid freezing method. So simply you are going to take the vials, add your tissue and the cryopreservant, and then simply place them at -300 to -1000 degrees centigrade. Okay, in this case the survival rate may be low because of again the shock created to the cells. From the normal room temperature, you are transferring them to the ice cold condition. So in that case, because of the cold shock, some of the cells may lose their viability. Then coming to the third method, combined freezing method. That means here the step 1 and 2 are repeated. It is a combination of low and rapid freezing. First you are going to maintain them at low and then at rapid freezing, high. Again low, again high. So here it is a combination. It is continuous combination process where you can store in liquid nitrogen. Again here also the survival rate will be less when compared to both. Then last one is dry freezing method. So here dry freezing method, the cells are dehydrated. So here you are not going to use any live tissue such as endosperm or the germplasm. Instead you are going to use the seeds. So when you use the seeds, they are completely dehydrated. That means the water content in the seed is completely removed or exhausted. So you can have the dried seeds which can be stored for very long time. Is that clear? So this is the process of cryopreservation. If you come to the advantage, you can store them for indefinite period or time period. Then no contamination will be occurred. Here there is no chance for the contaminating microbes or the fungal contamination. And then the space required is very low because the liquid nitrogen tanks are very small. Right, you are going to store your vials, so very low space is required for the storage, and also the handling is done by only single person, so very less labor is required for this complete process. Is that clear? If you have any doubts, put it in comments and subscribe for further videos.